Welcome to all. So today we are going to discuss very briefly about a very important topic called tyrosine kinase receptors and its pathway. Okay. So it will be a very brief introductory session to understand what is this tyrosine kinase receptor and its pathway. Okay, so let me take one pointer pen. Okay, so basically this kinase is a protein which will phosphorylate the other proteins, okay. So the phosphate will be taken from ATP and it will be getting phosphorylated, okay. So this tyrosine kinase is a very important receptor in human cell, which will be used by the most signaling molecule in human body. Okay, normally it will be available in cell in the form of monomer, which will inactive in nature. Okay, so to relaying the signal, the monomer needs to become dimer. So this is and once it is become dimer, then the phosphate, because as I told, when it will monomer become a dimer, then it will become active, and then only it will do the phosphorylation, and it will take the phosphate part from the ATP, and then it will further attach with the other signaling protein in the cell cytoplasm, and this will activate the transcription regulation unit of the cell. So this only we are going to understand very easily. So first we will go to the structure of the tyrosine kinase. Okay, so as already I have told that the tyrosine kinase, it will be available in the cell in the form of monomer, okay, which is inactive in the state. So one part, so this part it will be outside of the cell, which is called a signal binding site. So this is the extracellular domain, and another part it will be in the cell membrane, which is called transmembrane domain of the tyrosine kinase, and it will contain alpha helix part, and another part, the longest part is this part, which will be inside the cell cytoplasm. Okay, and this will contain the tyrosines, which will do the relaying, the signal relaying action will be taken care by this part. Okay, so this is the simple structure, simple way I have told the structure of the tyrosine kinase. Okay, then what will happen, the signaling molecule, okay, it will come and sit on the receptor. Okay, This is the signal. This is the binding site. Okay, if you see the previous slide, you see this part is the signal binding. This is the signal binding site. So the signaling molecule, it will come and sit here, and then this monomer, these two monomer, it will come closer. Okay, and it will fuse together, and it will form the dimer. So monomer, it was in inactive state, and once the signaling molecule come and sit on the signal binding site, then these two monomer coming closer, it will get fused together and it will form dimer. Now this is active. Okay, this is the second phase. Now what will happen? This dimer, it will attach with ATP, means the ATP will donate the phosphate, and this dimer, it will get phosphorylate. Okay, so now with the help of ATP, the dimer, the unphosphorylated dimer, it will get phosphorylated. So as I was telling, this kinase is a protein which will phosphorylate. So this tyrosine, it will phosphorylate this tyrosine, even though it can phosphorylate other protein also. So first they will phosphorylate each other. This is called self phosphorylation, autophosphorylation. Okay, so this tyrosine, it will phosphorylate this tyrosine, and this tyrosine, it will phosphorylate this tyrosine. So total 3 tyrosine, it will phosphorylate 6 times, which will take the phosphate from 6 ATP, and then that ATP will release the energy and become ADP. Okay, now coming to the next phase. Inside the cell, there are inactive relay proteins. This was inactive, but once this dimer get phosphorylated, then this inactive relay protein, it will come and join in the phosphate binding domain of the dimer, and then this inactive relay protein will become active, and which will activate different kind of cellular response. Okay, basically what will happen now, this inactivate relay protein once become activate, then it will interrupt different, then it will go to that signal, it will go to inside the cell nucleus, and it will interrupt, it will trigger the transcription regulation unit, and it will change the gene activity. You know, transcription regulation unit is the basically this will regulate the conversion of DNA to RNA. Okay, so if this kind of response is activated, then DNA, it will be converted to RNA, and entire the gene activity of the cell, it will be interrupted. So this is very simply, very simply, this is the tyrosine kinase pathway, and from here, you know, the cancer, basically this is the abnormal growth or abnormal cell division taking place due to this transcription regulation unit activation, and this tyrosine kinase is triggering that. So this is the basic concept from where the cancer drug discovery started. So this entire pictures I was searching, because I was thinking this is a very interesting and important part of biochemistry, microbiology. So I was reading the books, so I thought that it should be very simple way, the explanation should be very simple way, so that the common people also can understand. So this picture I got from the internet this all the pictures, and I will share the link of this pictures in this video description. And here on this lecture series will continue, and we will learn in the depth of this pathway and its inhibitor, how it can be inhibited, so that gene activity interruption can be controlled. All these things we will conceptualize in the subsequent lecture. So hope this will be useful to you all. Any question about this, you can ask me. My contact details, it will be given in the video description. Thank you so much. Thanks for your patience.